Hello. How are we doing? Are we ready to start receiving messages and viewing live streams? Again, please ask me your questions. I'm very excited to answer them for you. Yeah, that looks fine. We'll just leave that. YouTube looks fine. Cool. I have my unified chat here. I'm ready to go. Let's just have an explore of the map in the meantime. This should be displaying a bit better. How's that look on the live stream, by the way? Let's have a look here. Excellent. Let's try make the camera a bit smaller if we can. There we go. Okay. We can see most of the map now, I think. We'll put this up here. Yep. Nice. So America started reporting town by town and city by city rather than just state by state. So you can see that things have gotten a lot more serious across the USA. Which is really, really concerning, because these are just the numbers that we can see that have actually confirmed tests. And in the USA, they're not testing people who are asymptomatic. You have to have serious symptoms to get a test. Now, across down here in Australia where we live, it's not reporting town by town, but don't let that confuse you. There are absolutely cases in southwest New South Wales. It's just, it looks like it's only in Sydney and Canberra because we don't have the same discrimination in our testing. Oh shit, we're dropping frames already. This is not good. Mob crush is missing information. Okay. Oh well, that's not a problem. We're not actually losing frames. That's okay. So 818 in New South Wales. Let's see if we can look at Australia as a whole. Actually, can we? Australia will be 2,300 cases in Australia. Jesus Christ. All right. Perth 175. South Australia 170. Melbourne 411. Canberra 39. New South Wales 818. Queensland 397. And Tassie is now up to 28. And they thought that they were safe down there, but unfortunately no one appears to be safe from this pathogen. So what about New Zealand over here? New Zealand 155 cases. New Zealand, of all the countries in the world, is probably doing the best, both because they have the money to buy tests to compete with other countries, and because they're a little island very, very far away from everywhere else, even pretty far away from Australia. So, now we've got a number of cruise ships floating around the ocean right now. It's a French cruise ship. Really unfortunate if you chose this moment to go on a cruise, to be honest. Now, are any of these chats working? If someone is there in chat and able to talk to me, please say hi. Just so I can test my unified chat, whether you're on Facebook, YouTube, or on Twitch. Now, you've probably all seen this map, but let's see if we can find the USA temperature heat map. USA fever map. From wireless. Hello, Jody Bussell. Thanks for tuning in. If you have any questions, please feel free to ask me. If the audio is a problem, please let me know. Wireless thermometer. Try to remember the name of this brand. Kinsa map. Here it is. All right, so this is kind of cool. This is not diagnosing people by actually looking for the pathogen. This is using big data from thermometers to be able to observe where people are currently sick. So here you can see the trends of where people are generally sick and not sick. And here you can see atypical temperature readings across the USA. There's no data for anywhere else. That's why I'm using the USA rather than another country. Chat test 1, 2, 3 from Facebook. Excellent. Facebook appears to be working beautifully. Well, just if anyone is on the Twitch channel or if anyone is hanging out on the YouTube, which you I might try and post some links to those somewhere at some point. Please say hi there as well. But looks like the unified chat's working for those two. A little bit worried that Twitch is not working right now. So if we're looking here at the atypical temperatures, Florida looks bothersome. Now, this might just be because there's more thermometers down in Florida and more data reading, and that's why the other states aren't looking nearly as bad. But around the New York City, New Jersey area, around the Florida area, over here in Arizona as well as up here in, Jesus, I'm so bad at them, north of Oregon. What's this one called? WA. Someone who's an American expert, tell me. Washington? No, Washington's over there. I know who will tell me. This map will tell me. Seattle is in the state of Washington. Oh god, it is Washington. Okay, that's just confusing. God damn it, America. Yeah, so Washington, as we can see here on the actual sickness map, Washington 1,170 cases. Florida. Can we go to USA state by state? Yeah, Florida, 1,467 confirmed cases. So you can basically add up the next 20 states and they add up to the amount of infections in New York City right now. So New York City is obviously the center of the outbreak. Now, where else can we look? Because I think that's enough focusing on America and on Australia. Let's have a look at Europe. And it looks like the map's frozen now, so let's quickly refresh it. Well, that's wrong. 141,000 cases. That's a couple of 100,000 off dashboard. Are you including, or is that? Maybe it's just a bit laggy. It doesn't appear to be loading data from Europe right now. This is pretty slow for the job. Yeah, that's reading off the wrong number right now. We can see more cases in the first few than these all combined. Yeah, it appears to have stuffed up. Maybe I need to just stop putting so much pressure on my own PC's RAM. We'll close that. Close that. Let's try again, John Hopkins. Come on. Actually, you know what? Maybe if we just go back to and load the map from scratch. No. Okay. Apparently, John Hopkins has decided that reporting on Europe is too expensive, and they've actually just cut them out of the equation. So if we look down here, you can actually see the amount of global cases is just over 400,000. So 147,000 is clearly very wrong. But yeah, no data for France, no data for Switzerland, no data for South Korea. All the African data has disappeared. Ah, dear. Oh well, I guess we get away from this then, since that's no longer working. Let us instead peer into our lab archives and continue to plan out this experiment, as well as continuously check our email to see if this package has arrived. Nope, nope, nope. Nothing on that inbox. Check the other inbox. Alright. So, very, very clear what I need to do for the first while. After that, we then need to decide what we're going to be testing first, and that's really going to depend upon whether we've got a positive sample into the lab or not. We have a couple of positive controls on the way, but we also have potentially access to the Garvan Institute later on, where we might go and do the experiment there. Whatever turns out to be the most efficient. God damn, it is hard to breathe inside this mask. Alright, yep, nothing, nothing, nothing. Yep, I guess we just keep waiting then. So, it's going to be a very, very small volume reaction, just 24 microliters, plus the sample. And then you just give it a little spin or a tap on the side, and then you seal it and incubate at 65 degrees for 30 minutes. And that's the entire test. We have a tacit plan in place to work with the New South Wales Health to implement it in the drive-thrus, but that's just going to have to all depend upon whether we succeed in this first test. And I got to say, normally I'm very cautious in my optimism, but I feel very confident in this one that will work. And that's mostly thanks to the fact that we're using someone else's research. Oh, forgive me for speaking slowly. It's very hard to breathe in for each sentence through this mask. Okay. Let's go have a look at the papers, shall we? You can find the papers by going onto here and then opening up these hyperlinks on the primer sets. I might try and make it more clear exactly where the papers are later on. But they're also, you can also find them down the bottom of the page in the references section. So this is page 1, and we're actually now friends with Jin Zhao Song. And I've only had a couple of emails back and forth with him, but I'm very, very excited to have one of the original authors helping us out, because that's huge in the science world. You know, we are just a little lab, and it's really kind of him to actually hit us up. I won't leave his email on the screen. So let's get the PDF downloaded and have a look at it. All right. A single and two-stage closed tube molecular test for the 2019 coronavirus. Now, what does it mean by two-stage? Two-stage isothermal method dumped pen ramp. Pen ramp to enable high-level... But Sarah tells you discovered that in many cases, our two-stage is 10 times more sensitive than LAMP and PCR. Okay. So you design the LAMP primers. Since doing LAMP was carried out with 10... Yep, cool. These guys did 10 microliters of reaction mix. RNAs the coronavirus from different genera. Cool. This is what they compare against. Closed tube pen ramp. Pen ramp is 2 isothermal amplification processes: recombinase polymerase amplification (RPA) and LAMP. Carry out the RPA process in the cap of a tube and the LAMP in the tube. The RPA mix includes 480 nanometers of each LAMP primer, 1 times rehydrated basic buffer, and MGAC. The sample with various target concentrations inserted in the cap together, mixed together. RPA volume of 5 and 45. But a centrifuge not available, we decided to increase the LAMP volume to 70. That's what I was thinking. Wow, okay. So these guys have actually made it a bit more complex than we planned to do it. Hello, Uncle Bill! Glad to see that YouTube is working as well. Thanks so much for tuning in. Uncle Bill, are you able to see us on Twitch and comment on Twitch as well, just to check for me? I hope you are well and your family is well during this difficult time. So that's the 19 LAMP assay, and this is the RAMP assay, which gives you an even faster result in some results. Okay, so even with the 7, okay, couples per reaction threshold min. Yeah, another very important thing here is that there's 0 reaction gave nothing. That means that there's not likely to be a false negative. The main concern is whether... Oh, sorry, there's not going to be a false positive. The question of a false negative is still open, however. The limitations of LAMP, PCR, and pen ramp are 70, 70, and 7. Right, so what this is saying is that the ramp reaction, which we're not actually using, didn't get the transcode on Twitch this time, and my bandwidth sucks. No worries at all, Uncle Bill, no pressure at all. So, it looks like ramp is something we should investigate if we can get the LAMP to work, because it does appear to be able to detect a full order of magnitude less virus, which might be able to tell you someone who's infected a few days earlier than otherwise. So... But nonetheless, the LAMP did do its job. It even amplified the 7, so I'm sort of unsure why this... Okay. Threshold time. Okay. For the patients with low viral load due to insufficient detection sensitivity to RT-PCR, many false negative results and so-called relapse after negative cases are reported. It is likely that pen ramp can be used to alleviate the situation. So, this is definitely something we're going to have to look into once we get the LAMP working. It's a slightly more complex procedure on the ground, and so I'm not certain it's going to work perfectly for people working in the drive-through testing facilities, but it certainly looks like it would be promising for pathology labs to work in. Minimally invasive sample collection is desired for home and field testing. Nasal swabs consistent with common sampling method. Okay, in the absence of clinical COVID samples, we contrive samples by spiking and activating HIV viral particles in the nasal swab. Oh. We measured the amplification threshold as a function of sample incubation time, incubation temperature, determine the incubation for 15 min at temperature above 65 enough to lyse the virus. Incubation for over 15 minutes at 65 degrees is enough to lyse the virus and suppress inhibitors. That's kind of cool to know, even if you're not someone who works in this field. Okay, the rapid sample preparation may not be adequate for quantification, but is good enough to confirm infection. 100% sensitivity at 700 viral particles per reaction, and 75% sensitivity at 70 viral copies per reaction. So, that's not fantastic, but that is still pretty damn good. You don't want to see 75% sensitivity as a general rule, but it's better than nothing. And the 100% sensitivity once you get to 700 viral particles, which, when you're talking about things this small, 700 viral particles is not much. It's very, very tiny sample. It's just that, breathing in, if someone is infected only recently, the amount of viral particles in their mucous membranes might be quite low, and so the test has to be very, very sensitive. And so if it's 75% 70 viral copies, that's something I want to remember. Okay. Here's an actual diagram of the sensitivity. So yeah, here we go. But this is on par with the gold standard. Keep this in mind. The lamp that we're doing is on par with the gold standard. Ramp is even better, according to our author song. And look here. So they do the reaction in the lid of the tube. The lid of the tube, and then they flip it over and mix it together. That's actually bloody brilliant. I'm so genuinely impressed. So it's just, we need some RPA to try this out though, so I'm going to have to investigate that. And this is still a home test, so that's what a negative looks like, that's what a positive looks like. Very, very clear distinction. Okay, so this is the part which we sort of have used. In the conclusion, we figured out the primers that they found of the best use to them. Actually, sorry, that's the other paper. That's the other paper. So this is just them summing up their research. Although much simpler, LAMP test performs on par with the current PCR. The pen ramp is two-step, but it is even better. Alright, so that's pretty cool. Let's have a quick look at primer set 2. Now, I am curious whether Twitch chat is working. If you're chatting to me on Twitch and I'm not responding, it's just because there's a bug in the chat. So let me see if I can get that working. But thank you everyone who's following and tuning in. Alright, so primer set 2 and 3 are from this second paper. I think this is it here. No, it's this one here. Nope. It's this one here. Okay. So, these are the authors here, guys. Big credit to all of them. This is their work and not ours. New England Biolabs. These are the guys sending us the kit. Big thanks to New England Biolabs for their work with their kit, and we're hoping to be able to set up some kind of supply with them. Now, details. We all know about what coronavirus is. So, they created 5 sets of primers. These are 5 different experiments to try and see which primers are going to be the most effective. Hello, Sammy. Thanks so much for tuning in. Great to see you. So, yeah, let me know if you can hear me through the mask. I know I sound a bit silly and a little bit nasally. So let's get down to the primer discussion and where they actually compare the different primers. So here's the actual sequences of what they're doing. Let's pull those up in higher detail though. So this right here is the annotated COVID-19 gene sequence. Let's download that and open it up. So this right here is the molecule that has brought the world to its knees. It is 29,811 base pairs long. If we go in here and we look at it, it's just a bunch of letters. A's, C's, T's, and G's. Deoxyribonucleic acid, individual bases all tied together. 28,900 of them. And that's all it takes to hijack your body and turn it into a virus-making machine. Now, I'm not doing anything illegal by showing you this information here. Normally the sequences of viruses are more difficult to find, but during a time of heavy research like right now, these are very, very simple to find. You can just go onto Google and type in BLAST or BLAST gene search, and it's going to help you come up to a big database, and you can find this as well as all of the different genes involved. So, this one here is the ORF1AB protein. This is the one which we're going to be testing 2 regions of to see if we can detect the coronavirus. The first region, and just keep in mind that these little things are only 250 base pairs long. They're actually longer, but this is the only way I can really clearly describe it without having to make it look really weird. So, in this area over here, it is a slightly longer, it's about 269, not 250. That's where the first one's going to be. The next one's going to be the other end of this protein, at the tail end. And the final one is going to be over here in this nucleocapsid phosphoprotein gene. And it looks like this one over here. So let's have a look at these in a little bit more detail. First of all, the first paper, which I just showed you. We'll download this and have a look at it. So this is the first amplicon. I call it amplicon 1 because they don't actually give it a name, and the naming conventions in these papers are pretty weird. So these are the 3 primers that we're going to be using for the first test. These are the ones that have arrived right here. So you see these sequences on your screen right here. These are actually encoded in DNA, and then that's been frozen down and is in the bottom of these tubes. So we'll be putting those back into water today, and hopefully using them for the test to amplify this region between here and here. And in the process of amplifying that region, these nested primers are going to create dumbbell-like structures that will push a salt out of the liquid, and the indicator dye will then tell us that those dumbbell-like DNA structures have been pushed out, and that means that we've got a positive sample. Paper 2, this is the other paper. This is the other regions that we're looking at here. This amplicon, these are going to all look very similar to you now at this stage. So this one's 269 base pairs long, and if we go in here, we can see the same set of nested primers. They have different sequences, but the principle is still the same. Okie dokie. And gene NA primers and amplicons. This one here is 237 base pairs, so it's actually is about the same size as the one shown on the other one. And this one's in the nucleocapsid protein. So this is the amplicon here. It's this region, this gene. All coronaviruses will have this gene. So this so one of the questions is, can we distinguish between this coronavirus and, say, SARS or MERS? So once we get, I mean, considering there's not a MERS outbreak around the world right now, if you get a positive, it's probably coronavirus COVID-19. But because some of these features are shared with the other viruses, it's going to be very important to make that distinction and to know that this is indeed what we think it is. And so that will be an important part of the test later on to ensure that we actually are amplifying coronavirus and not, say, SARS. Once again, because coronavirus is everywhere and SARS is almost nowhere, it's almost certain that we would be amplifying the right thing. Okay. Now, I'm still waiting on an email to tell us that a package has arrived. When that package arrives, we'll come back here, get everything together. We will probably then take it across to a different lab where we won't be allowed to record to do the first test because we will be looking at actual positive samples. This is a PC1 lab, so it's not good enough for pathogenic samples. I want to make that very clear. We can't actually do the test on positive samples here. We can do the test on some double-stranded DNA, but because the test is designed to amplify RNA, and the first step of the test is to turn the RNA into DNA and then turn the DNA into a positive or a negative, doing the test here on the DNA sample is not going to tell us as much as going across to another lab and doing it, or actually testing people out in the field. So we ordered the positive control to look at here because it's important to at least know if it's going to be able to work in some regard, but that's not going to test all the variables. It's not going to tell us 100% this test works, and it's definitely not going to tell us the positive and negative hit rate, about whether or not we actually are telling all people who are positive that they're positive and all people that are negative that they're negative. Okay, so any final questions before I pop off for another few hours and head off to go see if this test kit has arrived? And also finish doing the maths here on the dilution calculations, make sure I've done anything right. Any final questions? It's been a pleasure having everyone here. As I said, we'll be trying to live stream regularly. We'll be live streaming on Meow's page pretty soon as well. So it was a pleasure having you all, and thank you so much for popping by, and I'll